Thanks very much, Dirk. It's really a pleasure to be here. Thanks again for the invitation. And I've seen talks in this sister lecture series from all the people who inspired the work that I'm going to present today. So thank you for organizing this amazing lecture series. All right, so I'm going to jump right in. And as you all know, our brain has this extraordinary capacity to adapt to the world at every stage of life. And that allows us to learn new behaviors and to adapt to changes that may come from within our body or from the outside world. And this amazing capacity is what we commonly call neuroplasticity. And neuroplasticity is particularly perceivable after brain injury due to drastic changes in cognitive and motor skills. But we also see wide differences across healthy older adults in how their cognitive abilities remain stable or decline as they age. So today we'll try to better understand what changes in the brain may explain these differences and how they may contribute to these cognitive changes. One concept that is commonly used to describe these changes in brain activity or connectivity after any disruption is cortical reorganization. But what is cortical reorganization? Cortical reorganization can be defined as like the remodeling of functional architecture of the brain, where changes in local processing result in a novel functional role for a given area. This is the definition that is given in this review by Mackin and Krakauer in 2023. But actually, in this review and in other studies, this concept of cortical reorganization has been challenged, and even its existence has been challenged due to inconsistent evidence. So in this presentation, I'll present 3 studies that use precision neuroimaging and neurostimulation techniques to address the following questions. Does the language network reorganize itself with age, and does it reorganize itself after a stroke? And if we can identify beneficial neuroplasticity mechanisms that support either the recovery or the maintenance of cognitive functions, how can we promote these mechanisms by modulating the specific brain networks that are responsible for better cognitive functions within individuals? And I'll wrap up with some future directions for this work. So as you probably have seen across studies, multiple hypotheses have been proposed in the literature to explain neural changes observed either in aging or in stroke at the network level. And in aging, previous studies have reported that we may observe reduced network segregation, which is when a network of regions that are specialized in a specific function become more connected to other networks responsible for other cognitive functions and less connected within itself. We may also observe reduced network lateralization when a network of regions showing more activity on one side of the brain in younger adults for a specific function may show more activity on both sides of the brain as people age for that same function. And we may also see increased recruitment of bilateral prefrontal regions with age, or reduced specialization of regions, which is also called dedifferentiation. And after a stroke, there's some overlap between these mechanisms, but researchers have also tried to identify which neural changes explain the recovery of different functions. And these observations include the increased recruitment of the hemisphere contralateral to the lesion, and the increased recruitment also of the executive control or multiple domain or cognitive control network. This one has many names. And in addition to that, the reactivation of spared regions that were specialized in the impaired function before the stroke also seems to play a role in stroke recovery. So, I'm not moving forward. All right, so all of this to say that we have all of these hypotheses, but it's still unclear which of these mechanisms are actually contributing to the maintenance or the recovery of behavior. and where exactly they are taking place. And the question we can ask is why a consistent framework of neuroplasticity has not emerged yet. And why is it so difficult to clearly establish a link between changes in behavior and changes in brain function, and specifically in some brain networks? There are many answers to these questions, and I'll present just a few important methodological points here. So first, when we look at networks that represent especially higher order functions like language, we can see using functional MRI that the regions involved in language show overall the same pattern of activation, but they also show slight variations in their topology across individuals, as you can see here across these 4 participants. And this is particularly important, especially in the frontal cortex, where regions from different functional networks are highly interdigitated. And here you can see 2 networks. In red, the language network, and in blue, what is called again the multiple demand network, or the executive control network, or cognitive control network. And going forward, I'll just call it the multiple demand network, or MD network. And this network is involved in executive functions. And while you can see that across individuals, we can find a similar pattern as seen in the probabilistic maps on the left, when we look within individuals in this inferior part of the frontal cortex, we can really see differences in the exact location of the regions that belong to each network. And these differences are important because when we average activation maps from fMRI data at the group level to do, for example, group comparisons, we may blur nearby regions that are functionally distinct into either the same region, and then we might lose power in how we estimate the response of these regions. And this may then make it more difficult to relate changes in a specific region or a specific network to changes in a specific behavior. Other reasons that may explain why it is difficult to disentangle neuroplasticity mechanisms, especially in studies looking at language recovery after a stroke, have been outlined in the great systematic review by Stephen Wilson and Sarah Schneck, where they showed that fMRI tasks that were used to investigate the neuroplasticity of the language network in stroke survivors used a wide variety of paradigms that may involve a complex set of cognitive processes. And these task confounds may make it harder to compare results across studies. So let's see how we can try to overcome some of these limitations, of course, not all of them, using precision fMRI to study changes in the language network, both in healthy aging and in stroke recovery. So the first part of this talk includes studies that I did as part of my PhD at Boston University under the supervision of Dr. Swati Kiran and commented by Evelina Fedorenko at MIT. And I also wanted to highlight on this slide multiple people from both labs who contributed to these projects. So first, what do we mean by precision fMRI? At least in this context, in typical fMRI analysis, we measure changes in both signal at each voxel, and then we average this signal at the voxel level across people, so spatially. And in precision fMRI, instead of performing these voxel-wise averages of individual brains, averaging is performed on the response magnitudes extracted from functional regions that are identified at the individual level. And this approach has been shown to be more sensitive to detect changes in activation in specific functional networks, and it leads to more accurate estimation of effect sizes, which in the end allow for more straightforward comparisons across individuals. And in the 2 studies I'm going to present, we focused on the language network, but we also examined changes in the adjacent multiple demand network to compare neural changes in networks responsible for different cognitive functions that may be affected differently by disruptions like aging or stroke, and also because multiple studies have hypothesized that the MD network plays an important role in language recovery after a stroke. Here, the language network, as we define it in these studies, corresponds to the core frontotemporal regions, supporting core linguistic computations that are related to accessing words and constructions from memory and combining them to interpret or generate linguistic messages. On the other end, the multiple domain network here has been shown to be involved in goal-directed behaviors and executive functions such as inhibitory control, working memory, or cognitive flexibility, and is activated across a variety of stimuli types. To define functional regions within individuals for these two networks, we use different fMRI tasks that we call localizers that have been well validated in previous work to activate these two functional networks. For the language network, we use a listening task contrasting intact speech to degraded speech. And we also use a silent reading task contrasting sentences to non-words, as you can see here, an example on the screen. For the multiple domain network, we use an arithmetic addition task and a spatial working memory task where people have to remember the location of squares on a grid. And each task includes a hard and an easy condition. And this contrast between hard and easy has been shown to activate strongly the MD network across different types of stimuli. And it's really showing this difficulty effect or cognitive load effect. So to answer the question of whether the language network reorganizes with age, we collected data from a large data set of young, healthy adults and from two independent cohorts of older adults. And in this first study, we used the reading task as the language localizer and the spatial working memory task as the MD localizer. I just wanted to mention, I don't believe that people who are 40 years old are old adults, but we had to set an arbitrary threshold here, and they're older compared to the younger. All right, so before I show you the results, I'd like to orient you to the plots I'll show throughout these two studies. And I will always show the results with young adults first, as you can see here, and then I will show the results for the two older adult cohorts, as you can see here. And the response magnitude plots will always show the condition of interest in a darker shade. So here, dark blue for the hard condition, which is the condition of interest, and the control condition will be in a lighter shade, here the easy condition in lighter blue. And the difference between these two conditions tells us whether there is a significant response to the task. And I'll start by showing you the differences we found between age groups in the multiple domain network, as most studies on aging have reported changes in this network. So here we found that older adults show lower response magnitude in the multiple domain network to higher cognitive loads. And this is consistent with the crunch hypothesis, also called the compensation-related utilization of neural circuits hypothesis, which describes that older adults may reach a plateau in terms of how much their brain can compensate for task difficulty and may show reduced activation only for higher cognitive loads. Second, we found that the multiple domain network shows less extensive activation with age, and these results were replicated across both cohorts. So this goes against the idea that the MD network would expand as people get older. And finally, when we look at synchronization between regions during naturalistic paradigms, we found that the multiple domain network showed weaker synchronization with age, both at rest and when people were listening to a short story. However, this result was only significant in one cohort of older adults. All right, so together these results show that the MD network seems to exhibit age-related decline across different measures of activity and connectivity. And this aligns with what we see in behavior across studies, because many studies have shown that older adults show a decline in executive functions. In contrast, when looking at the language network across age groups, we actually found that the language network remains highly selective for linguistic processing. In one cohort, it even shows a stronger response to the sentences condition. And these results are that I'm showing here are in the left hemisphere, but we found the same results in the right hemisphere, just at lower magnitudes. We also found that when looking at activation extent, the language network also does not expand with age, but it remains stable. And finally, we did not find any changes in the strength of synchronization between language regions as people age. Again, these results are in the left hemisphere, but were the same in the right hemisphere between hemispheres. And in addition, we also did something that I'm not showing here on the screen, but we did not find any difference in lateralization between groups. So overall, across these results, we can conclude that the core language network seems to remain very stable as people age. And in addition to looking at how each network evolved with age, we also wanted to see if they would show any sign of de-differentiation, or what we can, de-differentiation is when we see increased synchronization between the two networks, as has been proposed in previous studies. So here you can see the preferred responses to the task of interest for each network that I showed you earlier. I've already showed you this data. And next to it, I'll show you the responses to the non-preferred domain, which for the language network is the MD spatial working memory task, and for the multiple domain network is the reading task. And in both the language and the multiple domain network, we did not find any change in the response to the non-preferred domains. And these responses were minimal across groups or non-existent in the language network across all age groups. And you can see in the MD network that the contrast is actually opposite for the reading task compared to the language network. So this is the first piece of evidence showing that the language and the multiple domain networks remain robustly dissociated with age. A second piece of evidence for this claim is that we did not find any changes in the overlap between language and MD activation maps when using a standard statistical threshold. So here you can see for young adults that the overlap between language and multiple domain activation maps is much lower than overlap between two runs of the language task or two runs of the multiple domain task. And this remains consistent when we look at older adults across both cohorts, with a slight decline in the consistency of activation maps in older adults for the multiple domain network. The last piece of evidence showing a robust persistent dissociation between the two networks is that they remain strongly segregated with age. And we did not find any increase in the synchronization between the two networks in the first older adult cohort. And we found a small increase in the second cohort, but this is more important during rest than during story listening. So this change is probably not supporting linguistic processing. All right, so in conclusion, this first study showed that the multiple domain network exhibits age-related decline in activity and synchronization, which is consistent with, again, the decline that has been shown in other studies and in our study as well, where we see a decline in executive functions in older adults. The language network remains in contrast, very stable in older adults. It responds to language as strongly and selectively as in younger adults, and we didn't see any changes in lateralization or synchronization. And these results are consistent with the relative preservation of linguistic skills with age, especially for language comprehension. And in contrast to previous studies showing increased integration between networks with age, we found that the language and the multiple domain networks remain robustly dissociated in older adults. And our findings overall do not really support previous neuroplasticity hypotheses, such as the de-differentiation hypothesis or the hemispheric asymmetry reduction hypothesis showing differences in lateralization. So now that we know that the language network does not seem to reorganize that much in healthy aging, let's see if it reorganizes after stroke. And in this second study, we included 37 individuals with chronic post-stroke aphasia and 38 age-matched controls. And we used both the reading and the listening tasks to identify and investigate responses in the language network. And we used the arithmetic addition task to map and investigate the MD network. In this set of slides, I'll show you age-matched controls first and responses in individuals with aphasia second, as you can see here. First, when looking at the response magnitude within each network, we found that both the language and the MD networks showed reduced response magnitude to their preferred domain, which is probably a sign of persistent disruptions from the stroke. Despite this weakened response across the board, the language network remains highly selective for language processing and does not respond to the control conditions or non-linguistic conditions as much as language conditions in people. And there's no difference between people with aphasia and controls. And this is true across all functional regions of interest. In contrast to what has been suggested in previous studies, what we found is that the multiple domain network does not seem to be recruited to process language more in people with aphasia than in controls. You can see that there is no recruitment of the multiple domain network during the listening task, and there is this consistent opposite response to the reading contrast, showing that it is not specific to language. And this is similar to what is typically observed in healthy controls, as you can see here. And this is probably one of the most important findings of this study. Finally, we found that the language and the multiple domain networks show weakened synchronization within network, but remain strongly segregated after stroke, as can be seen by the absence of difference between groups in the synchronization between the two networks in the middle here. And this is during story listening, but we found similar results during resting state. However, when we know that people with brain injuries have different clinical profiles. And so even though we define functional regions within individuals, maybe looking at neural responses only at the group level is not sufficient to fully understand the role of these networks in post-stroke aphasia. And to investigate that further, we decided to see how these neural features predict language and executive function abilities in patients. And to do this, we tested the predictive power of 3 independent sets of neural features corresponding to activity and connectivity measures from either the language network, the multiple domain network, or connectivity between the two networks. And language and executive function abilities were composite scores that we extracted from a principal component analysis performed on a large battery of cognitive tests. And we controlled for lesion volume in our analysis. So overall, we found that higher engagement of the language network and higher connectivity predicts better language performance. In contrast, higher synchronization within the MD network during language processing predicts lower language performance. And finally, higher synchronization between the language and the multiple domain networks has different effects across hemispheres. It is associated with better language performance when it occurs in the left hemisphere, but worse language performance when it occurs in the right hemisphere. And in contrast, only multiple domain network features predicted executive function abilities, which is pretty consistent. So to summarize these two studies, we found that in aging and in post-stroke aphasia, the language network remains selective for language processing, and the language and multiple domain networks remain strongly segregated. In healthy aging, we found that changes in networks reflect changes in behavior, with a decline in the MD network activity and connectivity patterns, mirroring the decline that we can see in executive functions with aging, versus a well-preserved language system mirroring typical well-preserved language abilities and especially comprehension in typical aging. Finally, in post-stroke aphasia, both the language and the multiple domain networks showed disruptions and weaker response magnitude and within network synchronization, probably due to persistent disruptions from the infarct. And in contrast with previous hypotheses, we did not find that the multiple domain network would take over or compensate for language, at least language comprehension here in this study in any way. And these findings have actually been replicated in parallel to why we were doing this study in a set of 15 judged individuals with aphasia by Peter Declercq and colleagues recently, which suggests that these results are robust across independent cohorts. So there is still a lot of work to be done to further understand the role of these two networks in language recovery. And one way to causally understand and test their role is to use non-invasive brain stimulation. And non-invasive brain stimulation like TMS or transcranial magnetic stimulation could also allow us to potentially modulate activity patterns in these networks toward activity patterns that would be beneficial for recovery. And actually, a few weeks ago, Sophie Exparas gave a really nice lecture in this C-STAR series demonstrating how TMS can be used as a treatment in post-stroke aphasia and how it may improve language abilities after stroke. And this work is also building on the work of researchers like Margaret Mazer, Royal Hamilton, or Gessa Ludvigsen, who have published many studies showing the potential of TMS in aphasia. So just to give a quick reminder of how we think TMS works, it can be used to both inhibit activity in cortical regions, as well as to facilitate activity in cortical regions using different stimulation frequencies. And we expect that these effects may potentially distribute across the entire network, as has been shown in other studies using TMS. Okay, so this is the ideal situation, but in reality, targeting one single region or one single network, it can be pretty hard. And again, Sophie Exparas has shown in her great systematic review that TMS used in post-stroke aphasia most of the time targets anatomical or scalp-based regions. And in very few cases, TMS targeting approaches use techniques that allow to identify regions based on their function, using either fMRI or the direct effect of TMS on behavior. And of course, this is completely normal because fMRI is not that easy to use in a clinical setting. So we use what is feasible. But as techniques are progressing and becoming more and more accessible, we might be able to solve that problem of targeting a specific region more precisely. And the reason why it's problematic is that, as you probably know, one of the regions mostly used in TMS for aphasia treatment is the right inferior frontal cortex. And the problem is that the language and the multiple domain networks, as I have shown earlier, are located right next to each other in this region, as you can see here across these 4 participants. And I'm showing the left hemisphere, but this is the same when we look at the right hemisphere. And given that these networks have different functions and potentially different roles in post-stroke aphasia recovery, as we have seen earlier, how can we make sure that we target these networks with enough selectivity if we only use anatomical landmarks? So what is, and in addition to this inter-individual variability, if the best TMS treatment was to target the left hemisphere in individuals with post-stroke aphasia, how do we manage this additional inter-individual variability due to the lesion site if we use an identical target, anatomical target across individuals? So I'll quickly share some insights from a different precision neuroimaging approach that I'm using now in my postdoctoral work, looking at how we can selectively modulate or target adjacent networks within individuals. And the nice advantage of this approach is that it can be applied to any network and therefore to a variety of disorders. I'd like to acknowledge the contributions of all the people working with me in the lab of Dr. Randy Buckner and Dr. Mark Eldaev, where I can currently do my postdoc, and in particular, Randy's son, who contributed equally to the work I'm going to present. And I'd like to thank the NIH, of course, for funding all of this work. So in the first part of this talk, I showed you how we defined the language and the multiple domain networks based on fMRI tasks specific for each network. Here, we also define networks within individuals, but this time using resting state synchronization between regions and advanced Bayesian models. I'm going to go in a lot of detail on the models, but with enough resting state fMRI data, my colleague Jingen Du has shown that we can reliably identify all of the main networks of the brain. And as you can see here, the general network organization is the same across individuals, but each individual, again, shows idiosyncratic patterns in the specific location of each network. And now looking closer at these 15 networks, we can see again the multiple domain network now in orange. I'm sorry for the shift in colors, shift in labs, shift in colors. So the multiple domain network is here in orange and the long-range network is here in blue. And you can see again how interdigitated they are in the frontal cortex. We integrated this precision network mapping technique with a precision targeting pipeline developed by Lynch and colleagues that allow us to select the optimal target cluster for each individual within our network of interest. So that's the first step after estimating the networks, and then perform electrical field optimization modeling to prescribe the best TMS coil placement that will maximally engage our target network. And this takes into account the geometry of the cortex because, of course, the intensity might also not be as high when it targets the surface of the cortex or when it targets a sulcus, for example. And once we have determined the best coil placement, we can prospectively estimate how selective the stimulation will be at engaging a specific network by measuring the overlap between the hotspot of the electrical field, as you can see here in red, and each network. And you can see here on this graph the proportion of each network that is overlapping with the hotspot of the E field at different thresholds for this hotspot. And we can also measure the total and maximal intensity received by each network. And again, here you see that the target network receives the highest intensity of the stimulation, the highest intensity of the electric field. And as I said earlier, this approach can be applied to kind of any network. And my current lab works on TMS for depression. So our target of interest is a third network called the salience network, which for this talk, I'm going to show here a composite of the pink and the purple networks, also interdigitated with the multiple domain network here. And so using this precision TMS pipeline, we demonstrated that we can target each of these two networks with high selectivity above and beyond other nearby regions. And you can see that in each plot, the dominant region is the intended target. In addition to the selectivity, as it's measured in the previous slide, it's also important to consider, as I say, the electrical field intensity received by each network. And as you can see here, the modeling predicts that TMS will deliver stimulation at a much higher intensity to the intended targets and less so to the non-target networks. So these target networks will be exposed to higher electrical intensity. So this was just a sneak peek of a larger project, because beyond this prospective or retrospective modeling approach that we have developed, the next question, of course, is to know whether precision TMS targeting results in distinct behavioral and neural changes across the networks targeted, because it's nice to model how the networks will be engaged, but it's better to actually see if that matters and if there are differences in actually how the networks are changing, and if that changes behavior. So to do this, we bring people in, we scan them for about an hour, and we create from that hour of fMRI data an automated report of their personalized targets based on their resting state data. And we administer after that TMS to 4 distinct targets, so 4 distinct networks. And we then evaluate neural and behavioral changes after TMS by bringing them back into the MRI right after the TMS or by administering some cognitive tests. And another ongoing extension of this work is to examine if this precision TMS approach actually results in better response to treatment, of course, because we can see that there are some differences in neural response, but if it doesn't result in response improvement in a disorder, that might not be important for the clinic. So for this, we are collaborating with Andrew Crystal's team at UCSF to prescribe personalized TMS targets for each individual with depression who comes to the clinic based on their fMRI data. And we will evaluate after their treatment if the prescribed targets lead to better results in terms of TMS response than the typical standard clinical targets. And finally, in order to make this approach accessible for people with brain injury, we first need to adapt the first step of this pipeline, which is the precision network mapping approach, to brains with lesions, because as with every fMRI tools, all the tools are first developed on healthy people, which makes complete sense. And then we need to see how we can adapt them to brains with big holes in them. So now I'm collaborating with Jing and Du to make this possible and hopefully available to all of you soon. And what is important with this approach is that it's based on resting state data, so it doesn't require the active participation of patients in the scanner. So it's quite promising for people with aphasia because we wouldn't need to ask them to do much. All right, so that's all the results I wanted to show you today. You can look further into these studies with the QR codes here. And we wrote a review with Swati Kiran on neuroplasticity mechanisms. And there were really great commentaries to this review. So I really invite you to read that sort of debate between researchers who work on this topic, because it was really interesting to see these different perspectives. And thank you all for your attention. I'm really happy to answer any questions.
Anne, I think you can see the audience clapping here. Thank you so much for a wonderful talk. That was great. So much to think about. Unpack, yes. Awesome. So I'm going to start with to see if there's questions from our room. We have some questions online as well, but let me turn the camera and see whether we have any questions from our own room right now. Sarah, do you want to ask your question in person, or because I saw you had a great question?
So I kind of had a two-part question. My first one was in the earlier studies where you look at the functional activity with aging. I was curious to know if you accounted for accuracy, or even like if they're listening to those passage tasks, kind of their ability to hear and just hear those tasks. So like accuracy and hearing ability in that functional analysis.
Yes, that's a great question. So we didn't include it as like a regressor in our analysis. We definitely looked at accuracy for the spatial working memory task. And we asked people beforehand if they had any hearing impairments, and we excluded them if they had. Of course, there could be undiagnosed hearing impairments, so that might definitely play a role here. But yeah, we looked at accuracy for the spatial working memory task, and there was like a slight decline in accuracy for the spatial working memory task. So some of the decline that we see in the multiple domain networks are very much probably related to this decline in executive functions. And for the auditory version, so we use, for the language task, we actually use the reading task. So this was silent reading, and there wasn't any auditory stimuli for that. And for the study in post-stroke aphasia, there was also the listening task, and we also asked people if they had any hearing impairment. But again, like that could be undiagnosed, so that might be a bias here.
And then just kind of this is kind of a separate question, but I was just to throw it out there. I'm curious what you would think is in this task, in your task, they're meant to kind of be distinct, right? Like you picked tasks that are really good at differentiating these two regions, but in real life, that's not really how we function. And so you can imagine that sort of this reliance or plasticity may exist more in real life than in the tasks. But just to sort of speculate, would you think that if you were working with patients with aphasia, you were trying to teach them some strategies using rhythm or using music, do you think you would then see like this extension of functional activity into the MD regions or network more so than what you're seeing in this study, which almost kind of makes you think there is no neuroplasticity almost, right? Like there is no really compensation, but if you teach somebody a compensation, will you then see it?
Yeah, that's a great question, and I definitely want to emphasize that I'm not saying that there's no hope in, in aphasia recovery, because as you say, like we see patients getting better, of course, and we see like improvement, and I'm convinced that all of the strategies that we have our patients practice are really beneficial. I think it's two different questions in separating what we try to investigate when we say neuroplasticity. So the really specific regions that are involved in co-linguistic processing, so really like accessing meanings and combining words together, are these co-linguistic regions changing, and are some regions of the multiple domain network or other networks doing these co-linguistic processes? I think that's one question we can ask, and that's kind of what we try to ask in these studies. It's really does that cortical reorganization concept actually exist in the sense that some regions will change their function? So that doesn't seem to happen from what we have seen in our studies. Again, it's fMRI data, so that other more subtle processes might exist at different spatial resolution and temporal resolution. But I think it's a different question from learning new compensatory strategies and which require executive functions. So it's adding like sort of cognitive processes to compensate for language impairments, but it's not changing the brain. How can I say that? It's not changing some regions of the brain to process language like the previous language regions were doing. So it's really learning these new processes. And sorry, my screen just shifted. I just want to see you again. Yeah. So yes, I think I don't know if I'm clear, but I think it's really two different questions in terms of there is definitely new compensatory strategies, and I hope that one really good way to test that would be to actually, because this is cross-sectional, it's not perfect. We wanted to recruit people at the acute stage, but the pandemic hit. We couldn't see people longitudinally, but ideally, we would like to see if we do treatment with learning new strategies, does that reinforces the multiple domain network, which would be what we would hypothesize, because we would reinforce these executive strategies more. And so logically, we would see differences in the multiple domain network, and maybe this interdependence between the two networks would also change. I hope that answers your question.
Another question from the room?
And this is William Mashin. Thank you for the talk again. Very, very clear, very useful, I think. So I've got two questions. One's sort of a very big question, and one's sort of a minor question. Maybe the minor question is just, do you think there's still a role for traditional group studies that do voxel-based, highly neuroanatomically constrained mapping, right? So one of the concerns I have with functional localization, which people have been doing for a long time, is that there's assumptions about that those are in fact the same regions. If they're in different places in the brain, right, by definition, then that's a question. So do you still see advantages of the traditional group approach and saying like, look, we are localizing two voxels based on their precise anatomical coordinates? So that's a minor question. But the bigger question is maybe, you mentioned earlier, maybe you're still saying that there's hope for neuroplasticity, but I guess the question is, what kind of evidence do we want as a field to conclude that really we should be looking at other avenues, right, for treatment or support of people with aphasia? So, I mean, that's sort of a scary idea, but it could be true, right, that there really is not a lot of this going on. So then what do we do if that's the case, or how do we know when we're there? So anyway.
Yeah, these are great.
I have no answers to this, so please.
These are great questions again. I think for voxel-wise methods, I agree with you. I think like constraining within an MRI, for example, is still like interesting, and that's what we do. Like we still constrain our search within parcels that are based on probabilistic maps. And so if we can do a similar approach with voxel-wise averaging, if we don't have like a really like that's also anyway meaningful to, for example, develop new functional localizers. We need to first establish these probabilistic maps at the voxel level across people. So I think there are still avenues to use these methods. And also I think there is a balance to find between, of course, the resources that we have and what we can use across people. And especially if we want to develop fMRI for clinical purposes, there sometimes like that we need to find a balance between how much we can acquire in terms of data and the benefit for the clinic. So that's for the first question. And the second question, so you were, I'm just trying to remind myself, you were asking about is there a hope for neuroplasticity? Does that even like exist, and how?
Is there some kind of data that would tell us that we should really be redirecting our attention to other avenues, right? If those would be more fruitful and helpful for patients. So how do we know when we're at that point, right? This is pretty compelling evidence, right? And there's a lot of other data as well, but how much more do we need to stop or less redirect our focus in some way?
Yeah, so I think one thing that I don't want people to think here is because we didn't see like sort of like the involvement of the MD network, for example, during language comprehension. First, it might be different for language production. This is something we have not tested yet. So this is really definitely something we should look at, because language production might involve more of these executive processes. And second, I think what is important in these results is that we see that more activity in the language network is related to better language performance. So if we can modulate the spared language network to try to get back these healthy activity patterns, which we see in patients, like that they might have a decline initially and might like recover some of these activity levels at later stages of recovery. I think modulating these important neural activity patterns toward more healthy-like activity patterns is a promising avenue, especially with brain stimulation. How is that feasible? Which type of stimulation is required? There's a lot of work to be done here. But I really think it's important that's why like investigating which network is important for recovery would help us, I think, in prescribing more like precise treatments and specialized treatments and individualized treatments.
Thank you very much. Any more from our room? No. Okay, then I'll read out some questions we have online now. Kirana Tsapkini asks, does the lower functioning of the MD network in the elderly, so this refers to the earliest part of your talk, the first part, does that mean lower efficiency? I mean, does it correlate with worse performance, and in which tasks?
Yeah, we have, I think I've looked at the direct correlation. I have to go back because and I think it was correlated. I don't want to say anything not correct, but I do think it might be related partially to the decline in executive functions, and I think that's why we see this decline in activity as well. However, one thing we saw as well was that there is also this decline, at least in one cohort, during resting state and story listening, at least in terms of synchronization. And so it might not just be related to less efficiency, because even during resting state or story listening, which is not a direct MD function, we also see a decline in how the MD network is synchronized. So I think there are multiple factors here to take into account, and definitely future things to explore there.
Yeah, and just to tag on to that real quick, just as a follow-up question, in the fMRI experiments, how do you deal with accuracy? So are these errors and accurate responses collapsed, or do you only look at accurate, or does that affect?
Yeah.
That may also indirectly affect the bold response, right? Or its spread.
Yeah, and I think we should maybe do like a separate analysis just with accurate responses. We included everything. We had pretty high accuracy overall, like the easy on both hard conditions were about like between 70 and 80% or 85% of accuracy. So overall, we had pretty high accuracy, but I think it would be interesting to see if there are any differences when we look just at accurate trials.
Yeah, thanks. And another question by Tamar Truzman. Thank you, Anne, for a wonderful talk with 3 exclamation marks. So she really loves the talk. In your presentation, you mentioned aging and stroke together in the context of neuroplasticity. Given the MD network shows changes, which you refer to as decline in aging, does this suggest that aging-related changes are viewed as disruptions rather than natural trajectories? Additionally, following the findings of language stability in aging, when studying stroke patients, do you think we can compare their neural activity or connectivity to healthy young adults rather than age-matched controls? Thank you, in capitals again.
All right. Thank you so much, Tamar, for our question. I'm trying to fully understand both of your questions here. So the changes in the MD network in aging, does this suggest age-related changes as disruptions or natural trajectories? I'm not sure how opposite you think these two are, because to me, aging is a natural disruption, if we can say that. So I think in my mind, they're like pretty similar. And that's what is interesting, I think, to see across like aging, stroke, and TMS, is that I kind of consider all of these as different types of disruptions. Then of course, like we can compare healthy aging versus dementia, but I think it's more like a spectrum than a binary dissociation. So happy to answer further on that, Tamara, if you have a more like specific dissociation between the two. And the findings of language stability in aging, you said when studying stroke patients, do you think we can compare their neural activity to healthy young adults within the age match? Yeah, that's a great question. I mean, I still think that even if the language network response is stable, there are still other factors like vasculature that may be different between young adults and older adults. So I still think that it's much better to look at age match controls to avoid having these additional factors that may impact the differences that we see across groups.
Great. Thank you very much. Question from Tatiana Schnur. Hi, Anne. Thank you for a clear presentation. Please describe the rationale for using different localizers from the ND network between the stroke population and the aging group. For example, spatial working memory versus arithmetic tasks. I think this came up in your response.
Yes, so that was mostly based on the data that was available because the young adults had were like really collected as part of a completely different type of studies. It's a large data set that F. Fedorenko collected over time, and so they not as many people had the math task, the arithmetic addition task. So for this group, and same for the listening task. So for this group, we could only use the spatial working memory and the reading task. For the stroke patients, we tried to do the spatial working memory task, but the results in terms of task accuracy were not, we could not dissociate as well hard versus easy in patients. So I think we had to maybe fine tune a bit more the difficulty levels. And so we prefer to not use the spatial working memory task in patients to make sure that we had a good effect in terms of the MD cognitive load.
Thank you.
But just to add, sorry, to add to that, the other group in the Netherlands has used the spatial working memory task. I'm not sure if they used a sort of different timing or something, but they were able to find the same results using the spatial working memory task in patients with post-stroke aphasia. So it should be, I think, yeah, our task might not have been the most optimal one, but I think it's feasible.
Excellent, thank you. Sophie Alex Barra says, great talk, thank you very much, Anne, and has a question. To improve TMS accuracy, does the method you're presenting, which significantly enhances spatial accuracy, also assist in determining the stimulation amplitude rather than relying on the motor threshold?
Yes, that's a great question. So determining the stimulation. Yes, absolutely. It does. And it's actually a feature that is included in our pipeline, optimizing the dose. We haven't explored that in a lot of detail yet. We have a study going on. I'm actually working with a student right now to compare what would be the selectivity in terms of network engagement between using the motor threshold versus optimizing the dose for that specific target cluster. And there is a study done by Gesartwix and group actually who showed that there are differences between using the motor threshold or using more optimal modeling approaches, optimization modeling approaches. And so we are also going into that that set of analysis and investigating that. And I think it will be very important in the future. Maybe we will still need the motor threshold to have a sort of baseline and then fine-tune that level according to the specific cortical geometry of each network. And I think it will be very important in the future if we can, of course, if we have the data for.
Thank you. I'm just going to keep going because we still have a little bit of time. Another question from Kirana Tsapkini. Great talk. Doesn't the lack of MD network engagement in difficult language tasks go against the actual definition of MD and the way it was initially specified? So the implication is that actually hard language tasks should actually activate the MD network.
Hard language tasks should actually go against the actual definition of MD. I'm not sure
I think this may be a definitional question. I don't know if you can answer it.
Difficult. So I'm not sure how, with the lack of MD network in difficult language tasks go against the actual definition of MD. I'm not sure.
Yeah, I think the implication is that anything that is harder, doesn't matter the modality, should activate the MD network.
Yeah.
That's how I interpret the question.
Sorry, maybe. I guess if you were defining MD as domain general, by definition, does that include language?
Yes, I guess that's a good way to put it.
I see. Yeah. I mean, it does respond a little bit. We see that, for example, we see slightly higher responses to the reading task than to the listening task. not in the contrast of interest. So it's of course not specialized for language processing, and that's what we have shown, is that it doesn't change its function to be specialized in processing language, but it can still be activated. Of course, it's activated for any difficult task. So if we do a very difficult language task, it will be activated. What we showed is that it's not more activated in patients than in controls. And we see really this difficulty effect, for example, in the reading task between non-words and sentences, where it's more activated when we ask people to read non-words, which is more difficult, than when we ask people to read sentences, which is more automatic. So yes, I think there's no contradiction here.
Yeah, thank you very much. I had one last question, if I may. So, which also actually has to do with the point that William raised in his earlier question, with very specific to the precision fMRI more than the resting state data, right? So one challenge there is to show the intra-subject stability, right? So if you're now taking individual peaks of activation related to a particular task, and then say, okay, so that's the region. So how stable? So this is a general question, and I know there's publications on that, but how stable is that in healthy controls? And do you know anything about whether that might be less stable in people with stroke, which is a hypothesis I can imagine, right? That there's more noise in the system, so also spatially you might see more noise within subjects across trials. Do you know anything about that, or what's your...
Yeah. And we looked at that, like a dice coefficient between runs for each task, and I think, if I remember well, we did find lower dice coefficients in patients versus controls, but still, like, using the functional localizers, still high enough to have sort of reliable estimates. What is interesting on that question is, when using the precision network mapping approach that I presented for the TMS, the study that my colleague Jing Du has done is that they really showed how much data you needed to get reliable estimates of these networks. And I found that very interesting, because I think it's not that fMRI is very bad and like inconsistent and things like that. It's just that we need enough data, and that's a reality we have to face to have robust estimates of how these functions are organized. And we can have reliable estimates if we have enough data. Of course, that then have implications if we want to translate that to the clinic. But there is potential to have reliable estimates, at least, if we get enough data.
Right, but so that fact, though, that you need so much data to get reliable estimates, basically implies that there is a lot of variance there, right? Otherwise, if you had one individual, so that means you're essentially doing a group study within individuals, right? For each individual, you're doing a group analysis over the multiple different peaks, which eventually gets you one region, right? So in that sense, it's...
Yeah, so that was more like for the network estimation. For the functional localization, I think that the fact that we are constraining results within regions that we know in most people have a role in these functions limit the variability in terms of results that we can find, because that first, like, is in terms of power, that increases the power of our estimates. But it's definitely something I agree with you, Dirk, that we should explore further to know, like, exactly what is the reliability of these measures. And again, I don't have the numbers in mind right now, but I think we had relatively high reliability in terms of these activation regions, these functional regions, sorry.
Thank you so much, Anne, for a wonderful talk. And we're going to need to have you back next year or so to talk about the role of depression in aphasia.
Yes.
Thanks again.
Pleasure.
That was wonderful, and thanks to the audience for staying and for being here and for coming back in 4 weeks' time, December 13th, when we'll be listening to Ida Rangus. Thanks, everyone.
Thank you,
Bye-bye.
everyone. Thanks so much.